Hello, everybody. Please take your seats. We're getting ready for the final segment of the Canola Week show. Yeah, that's what I'm calling it now. Yeah, the Canola Week show. Welcome to the Canola Week show. All right, I'm just going to keep doing this until you sit down. All right, our last session is chaired by Alison Sunstrum, who you met yesterday on the Public-Private Partnerships panel. I always have to be careful how I say those P's. Anyway, I've met Alison, virtually a couple of times. We've talked before. It was great to finally meet Alison in person. Alison is going to, are you going to talk briefly and then? Okay, so Alison's going to give us a short address and then introduce the 5 people who will be presenting their startup ideas. Alison.
Okay, so thank you very much for staying. For all of you that stayed, I think this is going to be the perhaps the best part of the Canola Week. So one of the things that I want to talk about is just where I came from. I gave you a little background yesterday, and we started a company in a garage in 1990 in Alberta. And essentially what we built was a data acquisition and analytics platform. We were an automated phenotyping method, and so very pleased that Genome Canada started the same year we did. And we piggybacked on some pretty exciting genomic discovery. Our pride, we actually grew our company from cash flow from operations. What that means is we were never able to get money. No matter how hard I tried, there was no money to be had. But we exited our company. If you grow your company that way, you do grow hard, but you also grow very successfully because you're able to get customers and people to buy in to your technology. Now, what I was very fortunate for, we exited our company in 2019 to a UK private wealth firm. Now, that sounds very exciting, but that's not what I'm proud of. What I'm very proud of are the 60 or more universities around the world that use my technology. We're in almost every major agricultural university, and at some point, we need to be replaced. We need to be creatively disrupted. But from that start, we created about 7,000 publications. I went to ChatGPT to find out what actually we had done, and it was humbling. And there was more than 7,000 presentations. I tried to keep track of the number of grad students, stopped at about 70. Hundreds of seed stock producers used our data, thousands of researchers, and there was millions of animals measured. Now, what that actually created was an academic and economic spin-off of a little over $500 million US. Now, when you look at it like that, that's the value of a public-private partnership with a startup involved. And as I said yesterday, overnight success after 29 years of really hard research and development. Now, I did 2 things, and I mentioned one of them yesterday. I did 2 things when I retired. I took up golf, absolutely hated it. Lasted 1 week and then decided I had to go do something new. I didn't think I had another startup in me, but instead what I decided to do was look at mentorship. For all the number of things that I did, I made so many mistakes. If I had somebody that told me how to grow globally, I think I would have made less mistakes. The other thing that I looked at was access to capital, and I've shared that I could not find capital anywhere. The first thing I did on mentorship is in 2019, I joined the Creative Destruction Lab. And Creative Destruction really thinks about you take everything down that you think you know and reinvent. And the 2025 Nobel Prize for Economics this year was focused on the economic driver behind creative destruction. Now, creative destruction is actually not known by many people, and it started at the University of Toronto in 2012 and has grown around the world as one of the preeminent programs for science, deep science and technology, frontier science, and it's a Canadian success story. We're now in about 18 sites. We've expanded beyond this. And after 1 year with CDL, I realized we needed this in agri-food really badly. And I went to the team and I said, let's launch an ag stream. And they said, if you invest in it, we'll build it. So I invested, and I'm so very proud to be the founding partner of the Creative Destruction Lab agri-food stream. This year, we're expanding. In January, agri-food will go to CDL Berlin and CDL Doha. So from Canada, we're now moving all around the world in agri-food. The impact of Creative Destruction Lab. It's an 8-month mentorship program linking scientists, and I'm very proud. And I'm going to call on this, and he gets angry every time I do, but the grandfather of canola, Dr. Wilf Keller, is one of our eminent mentors, and he is just awesome. And many of the companies that you will see today have been mentored by Wilf and suggested to come here today. But the impact of Creative Destruction Lab, it has been more than 675 companies are participating this year around the network. 13 sites focused on global impact are operating right now. And the equity value that's been created by Creative Destruction Lab is $46 billion. That means the amount of money that was raised by companies. So it's an impressive program. Now, one of the things, we've talked a lot about innovation this week, but innovation is something that's used for everything from toothpaste to nuts. And despite decades of innovation, Canada is incredibly slow to adopt and invest in new technologies, and we are at the bottom of our peer countries in the OECD. We are just not performing at the level that we can. Now, innovation means 3 things. We had a conversation about public-private partnership, but innovation is really the whole process. It's from invention times commercialization, and then someone has to adopt it. Just getting it to market is not good enough. You have to have adoption. Now, why innovation matters, and I love the presentation on the return on investment of breeding programs. As an investor, I really want to see those kind of returns that we're seeing in Canadian research. Why Canadian research is struggling, I just can't understand it. We need to fund more. We need to be at the front. We need to be investing in frontier research, and we need to be investing in our startups. And it drives our economic growth. We're seeing our reduction in productivity largely because we're not investing. It builds resilience. It strengthens our global competitiveness, and almost every dollar we spend in research, no matter whether you think it's not worthwhile research or not, is driving at least 2 to 3 times, if not more. And AI in agriculture could raise productivity by as much as 30% by 2030. Now, venture capital is one weird business. I had to get involved, and I raised a $51 million fund to invest in deep science and tech. And it's not for the faint of heart, because every venture program or every venture company has like a 10-year span. Investing in many of your science and technology is going to take 12 to 14 years to get it to market. So we have to speed up and accelerate that path. Now, if you look at the potential productivity impact from different areas of research and different areas of investment, we invest about 10% of all our venture capital money into apps. So consumer apps that have a lifespan of a year, that actually brings no productivity back. In deep tech, we spend less than 5% on deep tech and research. Agri-food tech gets less than 2% of all venture capital dollars. Now, that's about the same amount of money as women get, just so you know. So if you're a woman, and if you're a woman or an entrepreneur of color, you really are behind the 8 ball. We got to change that. The future of food, transforming our food systems, could be a $10 trillion annual opportunity. And we're all sitting here talking about so much opportunity this week. I've just been stunned by the amount of opportunity that sits in this room. The areas that I invest in, and I add AI because there is nothing I invest in that does not have some attribute of AI involved. I'm also looking way out there. And I'm looking at quantum compute, and I've actually seen 2 investments that are agri-food and related to quantum. I mentioned yesterday, just because I really love this, and I did invest, and I have so little of this company that it's almost a shame to say that I'm an investor. But this company actually sets the tone for what comes next. We all want to be a unicorn. This is a true biology company that's a decacorn. And a decacorn is you got to have over $10 billion of investment, a $10 billion valuation, and they have raised a little over $550 million to date. Why it's really important and what's driving that investment or that valuation is the fact what they've built in their foundry. And I gotta say, I went to Steve Webb and Gifts this morning, and it blew my mind. That is the closest that I have seen to industry-related technology development. I was just stunned. So I've gotta say congratulations to Steve Webb, the Gifts, and the team at University of Saskatchewan for pulling that together. It's impressive. If you have a chance, please go and take a look. Now, I wanted to say what we need in agricultural technology, and I consider ag biotech very much part of that. We need translational pathways. That was the gap that was missing from our discussion yesterday. We need to be able to take technologies and products rapidly from lab to market. It needs to be faster, it needs to be clearer, the pathway is clearer, and it needs to be well funded. I have never been at a conference where I heard more about funding. That was every conversation through every presentation this week. Integrated capital. You need to have patient, risk-tolerant capital that understands science. And industry collaboration, co-developing with producers, companies, startups, and talent and infrastructure. You can't have a science company if you don't have a scientific staff. So it's so important that we're empowering students, labs, and test beds. Now, let's go on to the better part of the day. Thank you so much for your vision and staying here, and thank you to the Canola Council for this week. First up, I'd like to invite, we're going to try to do this in a rapid fire format. We're going to have 5 companies join us. I'm going to ask them to introduce themselves. I won't waste the time doing that. I know they can do a good job themselves. Some of these companies have come through CDL, so I know the founders really well. You have a cross-section of great founders that can drive opportunity in canola, in breeding, and through to value-added opportunities that will increase the canola market. So first up, if I could ask Renaissance Biosciences to join me on stage. We will ask each company to speak, and then we'll ask the founders to come back and answer any questions that you might have. So, John, please.
Thank you, Alison.
Yeah.
Let me just get... Hello and thank you. I'll just have the presentations put up and away we go. Great. So my name is John Husnik. I'm the Chief Science Officer and Office of the CEO participant with my co-chair, Maurice Boucher. We are based in Vancouver. We have basically focused on yeast bioengineering. We started the company about 13 years ago. We have about 31 dedicated employees. About 90% of them are all in the science area, and then we have a small admin team. And again, we will be talking about the use of microbes, particularly yeast, and that yeast would be Saccharomyces cerevisiae, and how it actually moves along. Why yeast? This is something that a lot of people ask us about, and I guess the first few things that you have to come to realize is its food grade safety profile. So this is the yeast that you make beer, you make wine, you make bread with. Saccharomyces cerevisiae, very simple, very well understood. It's got an excellent low cost of manufacturing at scale. So an important number, that is, if you can imagine those little packages that you get of yeast if you're ever making bread at home, to make that, it's under $2 per kilogram. Now remember that number, because it's going to become very important when we talk about application margins and everything else going forward. It's a robust, efficient microbe. It's one of the most widely used microorganisms in industrial microorganisms on the planet. And there's existing multi-billion-dollar markets and new large yeast markets, which we're also on the forefront of. So that's why we are interested in using yeast as our ideal microbe going forward. And again, now I'm going to just talk about, very simply, something about this marriage between RNA technology and yeast technology. Very simply, we're looking at double-stranded RNA at this time, and that's, again, RNAi interference, interfering with this information flow of DNA to mRNA to protein. And if you're targeting essential genes, et cetera, you can actually then harm a pest so well that you can actually kill it and then move on from there. So very simplistic overview, but this slide I'm going to spend a little bit more time on, and that is essentially this manufacturing process that's really quite unique here. So we have a base technology, a yeast, a proprietary optimized yeast. Anyone probably in this room very simply could go out and put a construct into any Saccharomyces strain and you'll get double-stranded RNA. With our technology, you get very high levels of RNA. We've looked at the genome, we've turned on genes, we've turned off genes, we've got a patent around this so that we can put and ask the yeast very nicely we put a construct in there and ask for a specific RNAi molecule, a bioeffector molecule, to be made at very high levels. When you have that, step number 3, you have a complete bioengineered yeast containing that RNAi target for production, and now it's very simply a simple fermentation. The fermentation that a brewer, maybe down the street at a small brewery, could use this yeast and make loads of it. It's the same concept. Simple sugar, a little bit of nitrogen, and you can get loads of this yeast, each of this yeast having trillions of molecules of this RNAi bioeffector molecule that you've asked it to make. The downstream processing is even easier than the bread making. Very importantly, it's inactivated, so you kill the yeast. So now it's basically a biological suitcase carrying these active ingredient, which is the double-stranded RNA molecule. So you kill it, you spray dry it, and now you have a product that's stable. Stable on the shelf, stable during transport, stable in all of these situations, and it can be sprayed. We've done tests across the board for tank mixes. It can last at least 3 weeks in the field, if not longer, before any degradation. So it's got a lot of unique attributes when it's inside of this yeast, this biological suitcase, as I called it. More interestingly, especially for this project that we'll be working on in the canola flea beetle, i.e. the crucifer flea beetle and the striped flea beetle, is that we've increased the amount of RNA that we're doing that because we're using now a VLP system. VLP is a virus-like particle that also can be made inside the yeast and have even a more effective delivery system. So we have a lot of great technology coming through this, and I'm just going to go quickly over some of those advantages. Again, RNA, extremely pest-specific. However, it is also very highly degradable. So inside of yeast, we kind of take care of that. It's still biodegradable, but now it can actually last in the field under UV and all other conditions for a multiple amount of weeks. As far as we've tested, it's gone to 3 weeks without any degradation. It's cost effective. Again, now we're making kilograms of this at $2 per kilogram, roughly, and that stage. Stable, as I've gone through, food grade, et cetera. And then we have the ability to do up to at least 4 different targets in one yeast. So you can go after 4 essential genes. You can even be mixing a yeast. You can go after different products, et cetera. So, and it's compatible, as I mentioned, with standard farming practices. Our first application, like many others, has been Colorado potato beetle. And the reason we went after that one, it's just easy to rear in our facilities. We built our platform on it. It was just a model. Instead of using what we had earlier, C. elegans, the worm, and Drosophila, the fruit fly, we moved into a more practical model. We're able to kill larvae at all of its stages, L1s, L2s, L3s, et cetera. We can kill adults. And again, it's very effective in the field, which has basically shown all of the same results that we've had in the lab to date. So we started working on that first, but we have a whole host of other insects that we're working on. So going very quickly, as I just said, the L1s, L2s, L3s, pupation assays, everything is very effective against this particular beetle. Even cumulative egg counts and average of eggs, everything we're doing, and I'm sorry if you see the words for our product there, it's not the one we'll be going with. The scientists have started with the product being called Peggy, then it went to Peggy Sue, now we're at Peggy Susanna, and every time we do a shift in improvement, we keep changing the name. We'll eventually run out of what we can do with the word Peggy Sue, and hopefully we come up with something better in the lab. So. With all of that based, what we've done to date, we have now an AFC Agriscience Program projects, eco-friendly precision, innovative RNAi encapsulation of flea beetle control. And this is where we're at. We started this just this year. We've been able to get a number of different crucifer and striped beetles into our hands, and we're looking at new potential targets. We've got 5 that we are confident will work very well. This is from a number of people who've been able to send us the flea beetles over to us. We're in the middle of doing the strain engineering right now, and then hopefully in the next little while we'll be able to give you some of the presentations and the results of that. So I only have 3 minutes. We'll just zip along here as we get through. Like I said, this would be very important for that VLP technology, virus-like particles. This now really expands us on how we can actually apply this product. All of the things that we're looking forward to, environmental benefits, economic growth, and then just the resilience, providing Canadian farmers with a durable, precise tool to combat evolving pest threats. And that's what this base technology does. It's going to be for a number of different insect pests and beyond. So I just want to quickly thank to all the people here, John Gavlosky, James Tansey, Keith Uloth, and Megan Vankowski throughout these regions of Canada that have provided us with the species that we've actually then basically did a complete genome sequence, and then we're able to find the five gene targets that we're interested in going forward, as well as acknowledge all of these people. Again, we're yeast bioengineers. We're not the experts in every single one of these insects, so we had all of these great people on this list that really helped us put this grant together that we'll be working and hopefully solving this problem for you in the near future. So with that, I'd like to thank everyone for listening, and I'll hand it over to Alison. Thank you, Alison.
Okay, thank you. I wonder if we could have the Corol team. Jonathan, I think both of you.
Good afternoon, everybody, and thanks for sticking around for us. We're Corol. I'm Jonathan Curtis, and this is Cynthia Ennie. We're a company that is going to make canola oil-based polyol. You might wonder what polyol is. I'll explain it to you in a minute. But first, if you'll indulge me, I'd like you to think of a scenario where you're in your field that's going to be planted with canola. You're applying a slow-release fertilizer pellets sitting in your tractor. You've heard that those slow-release pellets are coated with a polymeric material that's made from canola oil, and you're chuffed to know that it's going to grow canola. You sit back in your seat. Your seat cushion is made from, yes, you guessed it, canola oil-based foam. And it's a hot, dry day, and you reach for the chapstick, which is made from canola oil. So you get the idea. There's so many products that we can make from canola oil, And that's what we're about. We want to generate canola-based oleochemicals. So the story started when I was approached at the University of Alberta, where I was professor, running the lipochemistry group. And we were developing different kinds of oleochemicals from prairie oils. And I was approached by a fertilizer company who came up with the application I've just described and said, wouldn't it be great if we could replace the castor oil, which is a natural biopolyol, with canola, because we're selling it to canola farmers. So we developed that technology, we gained patents, and a few years later, we formed Corol. The details of this slide don't matter too much, except for the fact that polyols are a really big business. They're a $30 billion or so business globally, so there's huge potential. And of that huge amount, only a very small amount of biopolyol is actually made in the world. So our initial goal was to replace castor oil. So what's special about castor oil is that it is an oil that is hydroxylated, and 90% of the castor oil is one molecule, which is shown here, racinoleic acid. Now, it mostly originates from India. Canola oil, on the other hand, is grown in much, much larger quantities in the world. And it's obviously grown a lot here, so it's of great interest. But importantly, it's very unsaturated oil. That makes it very special for actually doing further reactions, because the unsaturation are what gives us a reactive site that we can then do chemistry to try and mimic the castor oil. So we developed a process that involves pretty simple chemistry. We want it to be as straightforward and as economical as possible. The product we make is 100% bio-based, or can be 100% bio-based in certain configurations. We don't need food-grade oil. We use minimally processed oil. And we add the hydroxyl group to make it look like castor oil or a polyol. So just to get back once more to the structures and our promises to the last bit of chemistry, the reason why the hydroxy groups are important are because if you want to make a polymer, you need some point of attachment into the molecule, and those hydroxy groups are that point of attachment. And the fact that it has 3 means there are 3 places where you can add a cross-linker to make a 3-dimensional network, and you have no loose ends. So we wanted Corol to look like that, and the structure on the right is kind of a complicated illustration of the fact that we can do that. And because there's so little unsaturation, the probability of one molecule not having at least 2 and more likely 3 or more is quite low. So we can mimic castor oil, which is this polyol material, but we can also do more than that because we can control that chemistry. So instead of having one substance, we can have a whole range of different amounts of hydroxylation. Castor oil, which is this natural biopolyol, is used in an enormous range of industrial products, particularly flexible materials, adhesives, sealants, paints. It's used in cosmetics, detergents. It's used as a polyol to make flexible foams, biofoams, and it's used as a lubricant as well, and many other applications. Just about all of those applications could be achieved using our product, Corol. So you can see there's a big potential market. So what we see as the future is to find applications for canola to develop more of an oleochemical industry than we currently have. If you look at the uses of palm oil and soybean oil, they are very, very large number of different products that can be made. And some of those are polyol products. Most of the polyol products, I forgot to mention earlier, most of the polyol products, I mean more than 90%, are petrochemicals. And we'd like to replace those too. So we offer what could become an industrial demand for unrefined canola oil, for which we need good supplies of canola oil, which you all have. We want to do this innovation in oleochemistry. We see a vast market for different products that we can make with Corol. And we'd like to contribute to the development of that oleochemical industry, but also to the development of renewable materials that don't rely on petrochemicals and don't require the canola to be used to be basically burnt as a fuel. And I'm going to hand over to my colleague.
Thank you. So as a startup, we've crossed over that threshold of showing commercial viability and having a minimal viable product, in that we've partnered with industry stakeholders, for instance, JOAT, a global adhesive manufacturer out of Germany, and they're US-based organizations. We've made adhesive applications from canola oil biopolyols, worked with Ford in making, as Jonathan mentioned, flexible foam applications for vehicles. We worked with Agrium Inc. back in the day and tested and demonstrated the use of canola biopolyols in slow-release fertilizer coatings, and then beyond that, coatings and even in lipstick and nutraceuticals or cosmetic applications. Why is this interesting or important? I've listened over the past 3 days in the conference, and so much work has gone into developing resilient canola seed, finding ways to reduce the cost for inputs for farmers so that canola is a viable crop. And this is a way for us to make canola oil a high-margined, high-valued commodity out there. If anything we've learned in the past trade war is diversification. I think innovation needs to lead to diversification, where you're not just selling to one or two major clients, but you have a lot of applications out there that you can use canola oil for. So, we've talked about the market validation that we've done in the past. We have committed LOIs, and the biggest challenge that we're taking on in the next year coming is scaling. We're looking at making our canola-based biopolyols on a large scale, and Jonathan and I, Tolib, and a few team members here today, please reach out to us. We've put a flyer on each table. We do issue out newsletters. We have a LinkedIn page. So if you're interested to know more, come talk to any one of us, and we'll be happy to connect you with more information. Thank you.
So if we could have Norm Janssen of Vivent please join us. As Norm comes up, Norm and his partner Carol proceeded through CDL. This was one of the most intriguing companies that we met.
Thanks a lot, Alison. Yes, we were not just practicing with CDL or starting the program. We were actually a graduate of the program, not through my hard work, but through Carol's. And just a bit about the program, if I can. It is it sounds like a mentorship thing where you just sit there and you listen and you learn, but it is a lot of work. It's not something for the faint of heart or those who think that they're going to just get by, and but they will challenge you. They will make you think about your various strategies and your product. And yeah, it was a great process. But what I'm here to talk about is Vivent. Vivent Biosignals is a company based out of Gland, Switzerland. The CEO, Carol Plummer, is originally born and raised in Banff. And they're basically signal people. And they started this company not knowing very little about agriculture, and that's kind of where I came in. And so that's kind of the start of the company. So where we're going, I think, with agriculture, we can go through all the early stages, and everybody probably knows a lot of those things. Right now, we're in a stage where everybody's talking about data-driven agriculture. What we think the next phase will be, particularly in the plant science area, is plant-driven agriculture, where the plants help us make decisions. First, it was in human health that was happening, where you can go to your doctor and he does an analysis and figures out what you need. Now we've then moved to livestock, where we've got monitors on livestock that can monitor temperature, estrus, a number of things. But we believe that the next phase will be plant-driven agriculture. And so what happens, and the principle of this science, plant electrophysiology, is that there are signals that move within a plant. And it is what we do is we listen in on those signals and understand, first of all, listen to them, get the signals, and then develop algorithms to translate plant language into our language. So here is an example of what I'm talking about with plant signals. So this is a modified Arabidopsis plant. It gets bitten by a caterpillar, and you can see a wave of signaling that goes through those leaves. And that is the plant saying that a caterpillar just bit me. And and the plant can tell the difference between a caterpillar bite and a thrip. So how it works, we connect a series of 8 plants depending on what you're trying to look at. And that is our 8-channel device. And we then give you you get to see it on a platform, whether it's on your cell phone or your computer, a dashboard that tells you how the plant is experiencing its environment, or as I like to say, but I get slapped a few times to say how the plant is feeling, because people don't want to know that plants have feelings. And this is our single plant device, which we use outdoors, and it attaches. We attach 2 electrodes in a plant, much like an EKG that if you have to go to a lab to get tested. And from that, we decode the signal, and there's a number of different areas where it can be used, and I won't go into reading this. If you want a copy of this presentation afterwards, just come up to me and we'll figure out how we can get you one. And potentially we can maybe put it on the site here with some of the other presentations. So, and what can we tell? What does the plant tell us is really the question. It can tell us general stress that it's experiencing. It can tell us whether it's thirsty, so moisture stresses, nutrient stresses. We have algorithms for 6 major nutrients, and it can tell us certain biotic stresses. We've got some diseases that it can detect, and also some insects such as thrips and so on that we have those algorithms figured out. I won't get into all of the science to it. think a lot of people's scientific brain is full from the last few days, but so where can we use this? How could this help the canola sector long term? So I'll give you some examples. So here we're looking at breeding drought-resistant coffee. A lot of the regions in the world where coffee is grown have seen major climatic changes, and so we can help companies with their research to develop drought-resistant coffee or more drought-tolerant coffee. You can probably see that research and development sign at the top. That's from Nestle, and that's who we're working with there. Other areas, optimizing irrigation. We've done a number of tests where we can optimize irrigation. In fact, we're looking at automated irrigation in greenhouses with some really excellent results. I guess the efficiency on that, this is an example, but of course, that all depends on how efficient the greenhouse was in managing their water prior to what we were doing. So this is the dashboard and what it looks like. So we can do this for optimizing fertilizer inputs into crops. How are you fertilizing? In a large part of the world, that's a concern because they're concerned about leaching. Feed the crop when it's hungry, if you can. So that is some of the things we do. Other areas. So different other projects that we've worked on or the potential to work on. So different stress tolerance between varieties, and that's the poster outside that we've done with the BASF, 4 BASF varieties, and looking at different stresses and their vigor on those different varieties. We've developed lighting recipes. Now that there's dynamic lighting, we can assist in developing lighting recipes for things like greenhouse crops. So a lot of interest in new berries, strawberries, raspberries in greenhouses, and how do they respond to different lighting regimes, different wavelengths of light. So early disease detection, we're working with the Cavendish banana with Dole in the Philippines on very early detection of fusarium within 36 hours after it infects a plant. And then they can come up with some sort of remediation and researching mediation. I think one that people talk a lot about is, that's been talked about here was weeds. And we always talk about trying to get the most vigor in crops, but how do we measure vigor in weeds and use that as to help us control them? So let's get them when they're weak, if that's what we need, or get them when they're vigorous, when some of these chemicals can work with them. We're doing a lot of work with biostimulants. How do they work? When do they work? And developing good recipes for that. And ground-proofing satellite or drone imagery is another area that we work. So really what I say is plants are talking. What would you do if you could ask a plant a question? What would that question be, and how could that help you? So talk to us if you like about what your challenges are, and we can see how we can help you. So the range of uses is wide. It's almost as wide as your imagination, taking it from research to demonstration right to a commercial stage where it can be used in commercial farming or greenhouses. Those are a number of the places where it can be used. So that's us, that's me. If you have questions, you can take down my email address. I'll be around for a little bit afterwards because we're off to another meeting at about 5 o'clock. Thank you.
Now, just so you know, I fall in love with every company that I work with. And Dr. Logan Skori, I met when he was a PhD student. He was presenting at a human health conference $100,000 prize was at stake, and he absolutely took the room. No one ever anticipated that an agricultural company would take that prize. So, Logan, Skori?
Thanks for the introduction, Alison. It's great to be here at Canola Week. My name is Logan Skori. I'm one of the co-founders and CEO of AgGene. We're a trait development company based in Calgary, Alberta, and we're looking at taking climate risk and turning it into genetic opportunity. For those of you who haven't met me, I am a farmer turned scientist. I had the privilege of growing up on a mixed farming operation near Canola, Alberta. We grow oilseed, wheat, and we also farm rocks. So we do live in a tough area. I was very fortunate to also spend some time working with Nutrien as a sales agronomist. And during that time, it was right at the time when BASF launched their pod shatter trait. So I really fell in love with traits, and I loved it so much that I spent the next 6 years of my life studying everything from gene editing to tissue culture, and really started the framework for AgGene. So AgGene, we're focused on trait development, and specifically we're focused on traits related to climate resilience, as well as improving the nutritional value of seed. So we work in canola, and we have a secondary focus in pulses. So again, working on abiotic stress for yield, seed protein for added value, and seed quality for enhanced sensory properties. So over the course of the week, I think it's been very obvious listening to farmers and agronomists that there are a couple key concerns. The first is the profitability of growing canola. The second is increasing input costs, and certainly on our own farm, we've seen canola go from our most profitable crop that we look forward to growing to every year to a crop that we're starting to scale back in our rotation. So certainly there are some major concerns, and a lot of this just comes down to yield. Canola is, as it was previously mentioned to, as a bit of a diva crop. I would also agree that we haven't seen really good yield improvements over the last handful of years. So in this graph right here, you see this green bar. This indicates the 5-year average of canola yield in bushels per acre. So you do see where there is an increase. This usually coincides with something like the adoption of hybrids, the release of new disease traits, and pod shatter. So really, as we look forward across the next 10 years, Canada's getting warmer, our summers are getting hotter, precipitation is sporadic, and winters are becoming mild. And this really is going to cause problems on the canola production front. So we really need to start thinking about, in addition to breeding, can we come up with solutions to help mitigate abiotic stress? And so at AgGene, again, there's a major problem. Canola is becoming a high-risk crop for producers. It's a large upfront investment. Guys are pushing fertility. Seed costs is dramatically increased over the last 10 years. And again, the diva crop does not like heat and stress, which coincides with full bloom. So at AgGene, we're really focused on a trait intervention to reduce the impact of abiotic stress. Specifically, when we look at the life cycle of canola, I like to think of stress as a fight or flight, similar to humans. Unfortunately, plants can't run away from their problems. So early in the season, seeding in May, when we get to early July, mid-July, we have full bloom. So what are some of the different strategies that we can exploit to mitigate stress? And keep in mind, this is not an exhaustive list, but these are the key priorities that we're looking at internally. So the first is on the flight side. So can we advance flowering time, and can we actually improve cold tolerance so we could potentially seed earlier and get flowering out of the hottest time of our prairie summer? And the second is, can we actually fight, and can we give canola plants active protection during a time of stress? So within our own internal trait development pipeline, we're currently advancing 5 leads for heat stress and flower timing. So we're actively deploying these as we speak. We've also made some advancements on cold stress. So we've got one trait in greenhouse testing. We're deploying another at the current time. And some of our earlier technologies focus on increasing meal protein and pod shatter. Some of these traits are already into advanced greenhouse testing. In terms of our strategy and how we develop these traits, we do not use we're not tied to a specific tool, so we have a very robust toolbox in-house. So we use tools like GMO gene editing, and we use these to really validate and assess traits. And using some of this information, we can then reverse engineer traits using mutagenesis. So this is a really important tool. I think it's underappreciated, but it's great because it does not have a regulatory bottleneck like a GMO trait or a gene editing trait for that fact. And for those of you that have been following the news today, the EU did just release that they've agreed to a preliminary framework on MBT crops. So this is a step in the right direction so that companies can commercialize gene-edited products. Our trait development pipeline is really enabled by our technical capacity in-house. So we've built a really robust canola transformation pipeline. We have our pipeline up to about 15% efficiency on transformation, and we can generate events in a few months. In addition to our transformation pipeline, we've also built a high-throughput screening platform. So we've built double haploid capacity, and we can use the double haploid process and chemicals to induce genetic diversity, and then we can use this platform to screen for different traits, so looking at things like abiotic stress, seed quality, and even things like different herbicide mode of actions. Our company is based in Calgary at the Life Science Innovation Hub, so we do have a world-class lab facility in Calgary. We're fortunate to have 1,300 square feet of private lab space and controlled growth space. So we are, again, very fortunate to be able to have low-cost and really high-quality lab infrastructure. Our team is a group of biotech, seed industry professionals. We've been supported by Canadian investors and look forward to continuing down this journey. We're very fortunate to also have a really good network of advisors that are kind of both technical and business. So they've been a real great addition to our team. Again, I would like to thank everybody for your attention, for the team inviting me to speak at Canola Week. And if you have any questions or want to learn more, please connect with me and let's have a chat. Thank you.
Now, finally, we have an add-on to the agenda. Curtis Cook from Insignum. And I just wanted to say that Curtis actually came based on just seeing this program. So that leads well into next year. So Curtis.
Perfect. Thanks, Alison. One clarifier, though, it's Chris, but we're close. Yeah, I can live with it. So here's what's kind of neat. I had the chance to go to Logan's lab, what was that, about 6 weeks ago, I think, during CDL session 1 that Alison was talking about. And he's doing some fantastic work. He's been great in giving us some ideas of things we can do to improve what we're working on as well. But Norm, where'd Norm go? There he is. Norm, as an agronomist, I wrote this down permanently on my hand, plant-driven agronomy. I love that. And I love that because between you and what Logan was talking about, it leads right into what we're talking about. So can you guys see what we've got? Perfect. So I've got 3 strikes against me before we even start. First one is I'm the last one up, but I think it's a good topic. Second one is I'm not from Canada, but I'm from Minnesota, so it's pretty close, right? Third one is I'm going to talk a lot about corn, but you're going to see why and how it ties to canola. So first off, and I loved it, Tony, I think it was Tony yesterday, one of the growers said it. I can grow 150 bushel canola, can't I? Can't do it profitably. Last year, National Corn Growers Association yield contest in the US, 491 bushel corn. Remember, I think it was Jason said, I know I can grow 150 bushel corn year in, year out. Who thinks 491 bushel corn is profitable? Absolutely not, right? As an agronomist, I can assure you, absolutely not. But there is that gap. And that's the gap we've been talking about the whole week, right? We know that the genetic capacity is there, but how do we start to mitigate some of the stresses that are in between there? And it's part of what Norm talked about. It's a lot of what Logan was talking about, right? Abiotic stresses and managing that. And we think we've got a pathway. So our founder, the CEO, Kyle thought a lot about this. He grew up on a corn and soybean farm, central Indiana, a couple thousand acres. And he thought a lot about disease. His older brother now runs the farm with his dad, but what amazed him is that fungicide application decisions, how we're going to manage disease, was made right now, in December. And whether or not you purchased it in December was based on your tax position. So you're either going to pay for it in December or pay for it in January, but you made that decision right now. And it made no sense to him. He thought there's got to be a better way. And so between the work he did as he was going to school, some of his internships, some of the other work he's done, he started getting this idea of, I think there is a better way. So we started with biotechnology, just like Logan was talking about, and said, okay, well, through biotechnology, we think there's a pathway there. You pair that up with precision ag. You pair that up with data analytics. Then you think about, how do you actually get it to a grower, right? How many people here have a cell phone, or especially a smartphone? Everybody, right? I even saw a few people even raise their hands. Everybody does. So how do you get it in a digestible format to a producer or their agronomist, like Jennifer yesterday, to say, hey, there is a problem here. Maybe you can't scout every acre, but we can help with that. So that's where we're at. So what Insignum has done is we figured out how to take anthocyanins. So in the top left picture, you see purple corn. Everybody's seen colored corn at some point in their life, right? You know that plants produce color. That's anthocyanins. Insignum has found a way to take that anthocyanin, in the case of disease we use purple, take that gene, attach it with a promoter, and move it over to where the disease infection happens. Why that matters is that buys you some time, right? So you think about that and think, okay, well, if today I have a disease that infects the plant, how many days till I start to see symptoms? The picture you see in the top right is a picture literally I took, again, with a smartphone, just walking through a plot one day, and I'm like, oh, look at that. Southern rust in the plant. That is not the rust showing up. That's the purple signal showing up before the rust shows up, to show you that you've got a problem and you need to be thinking about mitigation strategies. Should I be treating it? When do I treat it? Etc. So in simple terms, it really is as simple as this graphic. You simply put the trait technology into the corn, into the canola. You wait for the stress to happen, whatever it might be. In this case, it's disease. That then sends a signal via the plant, kind of like what Norm was talking about, right? Plants send signals. In this case, we're using anthocyanins. It turns purple, or sections of it turn purple. You can now, through data analytics, through technology, analyze that plant and say, yeah, it's time for us to make a decision. Here's the decision, and act before you actually see the symptoms showing up on the plant. So what are the results that we've seen so far? In 5 years of field testing, this being our 5th year, we just got the results back last week, we've seen between 7 and 10 days advance notice. This year it was 8.3 days to be specific. That buys you some time, right? Because now you can say, okay, well, what are the weather patterns? How bad is the disease? How long is it going to take me to get on the list? Or if I'm doing my own spraying, how much time do I have before I can get to that field? Do all the fields need to be treated? Because back in December when I made my decisions, I decided I was going to treat 70%. Maybe I don't need to treat 70%. Maybe it's 55% of my fields need to get treated. Maybe it's 100% because now I have real-time data analytics to make a decision with. The other thing that's interesting that we've seen with this is I've had this question multiple times. Well, yeah, okay, so you get a stress and it shows purple. It's specific to disease, and you can actually see that if you look on the picture on the right, you see where somebody's thumb is. It's actually insect damage on the plant. You'll notice there's no purple around that insect damage. It can tell the difference between I've got a disease problem or some other stress, be it hail, insects, whatever it is. So it's very specific to disease. So how do we come to market? Well, we've proven it out. I mean, like you saw, I can walk through the field with a camera and I can see it. That's not scalable. What we've done is we have done tests with drones, and we can see it within corn easily with drones. Now what we're doing is building an algorithm, right? So you could fly that field with a drone. You could get that data analyzed, and it could send you a report on your phone that says, hey, this is, you know, you're red, yellow, green. What are you going to do with grandma's 80? But it's time to make a decision. The way it works in canola or soybeans or tomatoes or potatoes, it's more likely more of the plant that will show up, if not all of the plant. So potentially, we expect you could actually pick that up with a satellite image. So you wouldn't even need to do it with the drone, just get your satellite images and look at where you're at. So again, it's back to using an algorithm and using data analytics and saying, okay, based on what we see of the purpling, it's time to make a decision, and or here's the decision we recommend. Or if you're an agronomist, even better yet, right? Now you can make that decision on behalf of your grower, because you're not just looking at the data from this, you're thinking about all the other things that are going on with their operation. So we really think there's great opportunity with this. I was thinking, I see Wolf, Wolf sitting over there on the side of the room, and I remember talking with him a few months ago about this. This doesn't need to be in every plant. On the corn side, you know, typically you hear about refuge in a bag. Makes total sense, just put it in the refuge. Why not do the same thing in canola or soybeans or other crops like that? It doesn't need to be every plant. It needs to be enough that you can use an algorithm to say, based on what we see, here's the recommendations we make. So where we're at today, corn, you saw the data, right? 5 years going. We consistently see that it works in the field. We see that 7 to 10 day advance notice. We don't see any yield deficit from putting that technology into the plant. It is cisgenic because it comes from the plant and it's just moved within the plant. We have our new scientists starting Monday that's going to be focused on canola. So we're moving into canola next. And then eventually other crops, soybeans, et cetera. We're also thinking about other stressors, right? So insects, nutrition, fertility, whatever it might be. We think that's also within the platform long-term as well. That's all I've got. Thanks, everybody.
So I'm going to invite all the founders to come and join us, and then we'll have questions. Chris, I'm really sorry. Curtis Cook is a head of an energy company. Really nice guy. Please come join us. I'll just stay up so everyone can grab a seat. Okay, do we have any questions for the founders? And you've got to have questions. And I'd really appreciate if you would come to the mics and ask questions.
Okay, so it sounds like we're a little shy online right now, so don't forget there's the QR code on the Slido, both for online as well as in person. But yeah, looks like we have someone up there.
Thank you.
Hi, Colleen Christensen with National Research Council, NRC IRAP. What I'd like you guys to talk about is field trials, small plots, field trial, commercial scale field trials. What do you do, and how do you access the site to do that? Do you do it through contractors? Do you do it through the university? Can you just talk about that aspect of
That's a great question,
TRL
Colleen.
7 to 9, that's what we're focusing
okay.
on, TRL 7 to 9.
Do you want to take that question first?
Sure. Not sure if this is on. Hello?
It's a slider on the bottom.
Hello? Yeah, I've tried. Okay, there we go. I mean, for us, what we've done is partner with universities and college, like Lambent College, and their greenhouse, and apply for funding and do tests there when we're looking at slow release fertilizer using canola biopolyols. And that seemed to work well with us. Though, you know, with educational institution partners, and going funding routes, it doesn't quite go as fast as startups would like it to go sometimes.
Maybe those that are really running some deep trials like Logan, or perhaps you'd like to respond?
Yeah, I guess in terms of field testing, certainly early on you're limited with seed quantities, so working in small plot and then slowly kind of scaling to larger plots. Certainly with a crop like canola, obviously containment and confinement is an important issue, so the best bet is to work with like a CRO. I know Dana's here somewhere. AgQuest is a great option. It's really important because you don't want trade escapes and causing issues throughout the industry.
Maybe somebody could speak to the cost of those trials, because for you don't have access to regular trials. I think that's the biggest challenge for a founder.
Yes. So for us, we did start our very first field trials. We went through PMRA and got the approvals, and then we worked with consultants in out east, PEI, as well as Manitoba. That worked, and it was relatively cost effective. However, as a small company, we did need to go a little bit larger and a little bit broader. And at the same time, we started working with a European company called Certis Belgium. They became a partner. They took on all of the field trials for us. So we were doing field trials in multiple sites in multiple countries throughout, I would say, France, Poland, Germany, etc. And now some of the pitfalls for that, again, is they own the data. So we were getting the results, but we can't use it to go forward. Other companies that we worked with, we were able to use the data. So we got the benefit of learning what our product was doing in the field and obtaining that data for ourselves for showing it to investors, et cetera. Whereas the other time, even though it was excellent field data, we couldn't share it with anyone else.
Wow, then that actually devoids the reason to do it. Please.
Yeah, so I have a technical question for Chris. So are you taking efforts to control gene flow? Like you say, it's a cisgenic event, but it's still an independent event. And how do you are you considering means or take action to prevent flow of that event into the host, into the surrounding crop? Because certainly in the Canadian context, that would be, from a regulatory perspective, critical.
Yeah, so that's part of what we do need to work through. So my background prior to this role was I worked for Syngenta for 26 years. And a big part of that was actually working around some of our crops that we did need to control where the product went, from our anagen product to agrosure and things like that. So working with the grain trade was critical. You can't stop cross-pollination and gene flow. You can manage it as well as you can. Logan, I think, brought it up, right, as far as...
Unless you had some pollen control gene within your...
Right, so part of the benefit is being genetically enhanced and genetically engineered, with it being cisgenic versus transgenic, so that will help with the grain trade and it being a concern. But it does need to be managed through stewardship programs, particularly early on. I mean, right now, and that was even part of doing trials, one of the challenges is a regulated status. It's relatively easy to find land to do trials. It's dealing with the regulated status of products and all the work that needs to go around that, the reporting and everything else. Taking that from a regulated status to a commercial status ties right to what you're talking about, and that's the gene flow and managing that crop, not just from making sure the planter's cleaned out, to making sure that the grain is being transported to where it needs to be, that it's being followed all the way through the process, even down to, and not limiting, but talking to your neighbors to let them know, I am going to be using a different product this year, and making sure that, if not through, logistics as far as setbacks from their fields, if there's restrictions on it, making sure that you can also look at other ways of pollination setbacks and things like that. But there are ways of doing it. It's not easy, especially early on, but there are very good ways that we've seen commercial products that can manage that.
Yeah, I think that'll be an important consideration for applications in canola.
Absolutely agreed. Great question. Thank you.
So, do you have another follow-on?
I have a second question.
Great.
Yeah, so it's with respect to the sensors. So the images you show of the sensors, they're all on the stem. Have you looked at placement of sensors on the adaxial surface of the leaf? And do you get any difference in signaling depending upon where you place your sensor?
Oh, good. Okay, I'm good. It depends what you want to measure and why you're measuring. But
you're representing as kind of a catch-all sensor. So
that's why we put it on the stem. It's in two places in the stem. So it's just like an EKG. You have a reference and you have a live. And so most of the stuff is done in two places on the stem. The odd research project, we've put it into the stem of the flower. And so if you want to do something very specific related to a flower, that's where we would put that's where we would put the main sensor, and the ground would be anywhere near the stem.
Do you do it on the leaves at all? Because if you're thinking about a drug response and stomatal closure.
We haven't done it on the leaf because we don't think we need to, because that message goes up and down the stem.
Okay. I'm not sure.
Great questions. There's an incredible brain trust in this room. And the things that founders need more than anything is stress testing. So I appreciate the questions. Jay.
Yeah, hey. Just a general comment. I don't know whether the 5 or 6 of you know this, but this is the first time we've ever done this at Canola Week. I love this. So Will
I love it
and
too.
Alison, thank you so much for putting this together.
Thanks, Jay.
I have questions for all of you. I'm not going to do that now. But we've got fewer and fewer big companies in agriculture, seed companies, the chem companies. But nobody knew 20 years ago that Tesla was going to be a major car company, or that Apple was going to be the biggest camera company in the world. So is your ambition to enter the market as a commercial brand or company, or do you want to sell it off to someone for billions of dollars? Both sound great, but I'm just wondering, do you want to come in and disrupt the commercial space with your own company label?
I maybe make a comment on that because we are VC backed. We just closed a $12.5 million round, and those VCs are wanting to make money on their investment, and they want to turn around and sell us or sell their investment to get good return. In return for that, though, we've got strategic investors that are helping connect us to market. So, we've had interest in buying. How much do we want to control who buys us? How much do the other investors want? Yeah, there's been interest.
Is there anyone on the panel that's building their business for growth, to have a business long term? Like 29 years?
Well, I'll try and 29 years would be great. We're not VC backed whatsoever. So we've been doing it with shareholders, and we have about 100 shareholders. Some of them have been in there for over 10 years. And so there is this demand to have some liquidity. So you're always being pushed a little bit from, at least in our case, some shareholders saying, I put some money into a private company. When do I get it back, kind of thing. And from my own perspective, to answer the question is, when I see what we can do, particularly with this VLP technology, I want to be the company that's best in class for that product, just be known for that. So yes, this is something that we do want, but you do have those pressures that, people have tried to buy us now 3 times, and we've always said no because they came in really low. And that's not fair to those shareholders, even though they want to see a return. But it's also not fair to the scientists, to the whole vision. So there is this kind of a push and a pull that you're fighting through this tide to actually get the best you think you can. And if that can be us being our brand, being best in class in the market, that's what we're shooting for. It's a hard road.
Yeah, it's a hard road. Please.
Yes, I really loved the presentations. As a producer, I don't always understand the science, but I certainly can see the benefits to many of the things that you're working on. I guess my question is to the Renaissance, is like, when will we be seeing flea beetle control? I mean, that's something, I'm from Manitoba, and it seems like every year we're fighting the flea beetles, and if we can get a product that will work for 3 weeks, people will be lined up at your door, day one, and we'll be wanting to use that product.
Excellent question, and exactly want to do that. And I think, and that's why we're here. We started off with the Colorado potato beetle, and we're looking into cabbage stem flea beetle with another group in Europe, and now with canola. But really, it's because we started. It is a huge product for Canada. It's something Canadian producers do very well, and it's a real thanks to the AAFC. We got a grant from them in order to actually do that. So we're working as hard as we can. I've got a great team of entomologists and molecular biologists, people that are much smarter than myself, and we are working diligently to get that. And we've seen how we've been able to do it for all these other beetle species and into lepidopterans and everything else. So we're very confident in it, but it will take us at least 3 years because it's a harder bug to kill than it is for, say, Colorado potato beetle, for example. So it's a tricky one, but I hope to see you in 3 years and give you the best-in-class product and be there in Manitoba.
I hope to be there.
Yeah, thank you.
Did you have a question from online?
Yeah, so we have some questions here, a lot of great ones, some technical, but one for Corol regarding the oleochemicals industry. What is the optimal fatty acid composition to make the castor oil replacement?
Is this on? Yeah. Great question. I would say we need fully unsaturated oil, or as unsaturated as possible. Beyond that, it totally depends on what product we're trying to make, because one of the key advantages of the Corol technology is that we don't just make one fixed polyol like castor oil, but we can make polyols with a range of different hydroxyl values and unsaturation and viscosity and general properties. So depending on what you're trying to make, you might pick a different oil or a different combination of oils. That being said, our go-forward path is primarily canola or canola plus one other oil.
Thank you. You have a question?
Yeah, I have two questions. Sorry. I have two questions, one from Renaissance and one from Chris, I believe. So the question about, so your yeast system, the example that you provided are for insect. So if you want to use it for pathogens, you need that your yeast secrete this, the RNA into the plant, or at least into the environment. So it is in direct contact with the pathogen. It goes through the plant and then it's fed to the pathogen. Does your system work for the pathogen?
So the way it currently works, the yeast, the RNA is going to be contained within the yeast, okay, and the insect has to eat it. So it won't work for all insects under the current way we are working, or say our generation 1. So it literally has to be eaten, and then the yeast is digested inside the gastrointestinal tract of the insect, and that's where the RNA is delivered. So what we have done now, the next step, and that's literally part of the canola flea beetle grant that we've received, is to develop this with the VLPs, the virus-like particles. So these are a lot smaller, and you can now keep it within the yeast and just have very high levels and do the same process, or you can extract. Now, the VLPs are not exported out of the yeast. They're contained within. but it's quite simple, and the art is known how to do it on how they make yeast extract all the time. Essentially, you blow up the yeast, and you actually have a semi-purified product that's a VLP, which is much smaller now, and you have much broader applications on how you want to do that. It's about 42 nanometers in size, carrying a payload of RNA now, and that potentially could be used for all kinds of different insects and potentially even microfungi or anything else, because now it's not a giant yeast cell, which is 10 microns, but a small VLP, which is 42 nanometers. And you can even decorate that VLP in order to target something very specifically. So we've broadened that, and this project will be one of the first places where we'll be using the VLP technology.
And that would be uptaken by the plant as well? Can it be?
We haven't done the studies, but it is a great idea because we think that can also be done that way as well, taken up by the plant.
Okay, thank you. For Chris, I had a question. So the promoter that you have, you showed that it doesn't respond to injury to the plant, if I'm right. But presumably it's something that it's responds to plant defense. So if that's the case, all the pathogenic and non-pathogenic microbes are inducing some level of plant defense. So how does it differentiate really that you know is a pathogen? And then even there are pathogens that are not really devastating to a specific crop like maize or canola. Or so they are say that there could be Fusarium graminearum on the field, on the canola field, that triggers some response, but it's not a pathogen of canola. How do you differentiate these signals?
That is a fantastic question for our founder. I genuinely don't have any idea. I'm just an agronomist.
Okay.
But we'll follow up. So if you catch me after this, let's follow up and I'll get you an answer.
Sure. Thank you.
Thank you.
Thank you. Great questions. Anything else online?
Lots online. I'll choose one maybe to Logan, so you can pick up a mic here. How are you solving the issue of competitive germplasm for your traits?
Yeah, certainly. Access to germplasm for startup is by far the #1 challenge. I understand that's a major investment that major companies make. This is something that you need to protect by all means. Fortunately, we have had some really good conversations with some seed companies that are going to provide germplasm. So, we want to be able to put traits in elite genetics so that we can test what the true impact of it is. It's if you use an older variety like Westar and you see a 25% increase in abiotic stress tolerance, well, that doesn't mean anything because it's 30 years old, 40 years old. So, we need to be able to deploy traits in elite parent materials. And this is a very, very critical thing that I think often gets overlooked in academia or even in the business world.
So we will go back to online for one more question, but I would like to ask everyone on stage, you've got a whole bunch of different resources in this room. While we're answering the online question, can all of you think about what can this room do for you as a startup? Did you have one more question online?
Yeah, well, we can do one more question, quick question. This is for John. For flea beetle control, you need to kill fast or stop feeding quickly. And I think this is in reference to some of your charts that after 5 days, then you start to see the mortality. Is RNAi suitable for this?
That is a great question, and the RNAi just technically on its own is a slower acting active ingredient. That's well known. The things with the VLP technology or even the yeast, we're looking at a multimodal action. So we're already looking at different things to go along with the RNA in order to make it faster and kill quicker. So RNA on its own, this is a great question. You're absolutely right. There's a whole series of steps that have to be done in order for that insect to die. So even if you have multiple different types of RNA attacking the insect, double-stranded RNA I'm speaking of course, and at high levels, there is that little bit length of time that it does take. Hence, making it compatible to other modes, all within the yeast itself, is what we're working on. So great question. Yes, thank you.
Okay, so thank you very much. Can we start at this end? And what do you need from this room?
I think what we need is some buy-in that the concept of developing or further developing canola oleochemistry is a good idea, because for me it seems like a great opportunity. You have the product, you were looking for stable markets. There's nothing you can do with soybean oil you can't do with canola oil or some version of it.
So you need market validation.
I do, yes. Yeah.
Okay, Chris.
Well, I think it's a great idea, so there's one for you. The same thing. That's actually, I was going to put in one last slide that said, if you want to provide feedback on your farm or as an agronomist or somebody in the industry, do we need technology like this? What's the value of technology like this? Would it help your ROI? That's the kind of things that we really want to get underneath, particularly as we're moving through CDL as well, to really say, here's what this could bring to the market and why it matters.
Okay, Norm.
I think much the same. As you think about our technology and the projects and applications for funding that you're looking at doing, if you want to talk to us and get a better understanding of how our technology may fit into that project or into that research project, demonstration project, or commercial project, let's talk and we can see how we can work together.
Cynthia, was that covered in that question, or do you have another ask?
That pretty much covers it. The only thing I will add is, partnerships, in these early days, it means a lot. So definitely reach out if you're interested.
Logan?
Yeah, partnerships. I know that canola biotech is not as attractive as corn and soybeans, but it's still a very critical crop for our farmers in Canada. So, don't be afraid to engage with startups. We're very lean, we're agile, we get stuff done probably faster than some larger corporations because we don't have the bureaucratic stuff to deal with. So,
And you don't have the money.
the money.
Please.
Yeah. As we are starting quite early, this is our first year working with canola flea beetles, our technology is really a platform. And all my scientists, they're either molecular biologists or microbiologists in all this area. We're not experts in this field whatsoever. So this is a great opportunity. Thank you. But we can go after other diseases. And and this is where I think this what I don't know. But sometimes you say something and I'm like, wow, maybe we can actually attack that with a yeast-based, because I always think yeast for everything, a yeast-based system to solve that problem. And I don't know what that will be, but if you have some, even if it's a little bit off the wall, come talk to me and we can say maybe there is a way to actually explore that and come up with something really new with your idea. Thanks.
Well, thank you all for just an amazing Q&A session. Jay, you look like you want to say something.
I just have to end the day. This is not on.
Okay, so thank you very much. Let's thank our founders for joining us.
Yeah, thanks.
And thank you, Jay.
Yeah, thanks, Alison. So I've got my three farmers. We're going to come up and close the day. Thanks to all six of you. That was excellent. Like I said, I will be following up with you. And John, I'm just thinking, like with Alan mentioned grasshoppers yesterday as a problem, and they often start in the ditches and then move into the crop. So you could have, and they go through a number of nymph stages. So it could be a slow-acting thing that could control grasshoppers in the ditch. Anyway, hey, here I am. Okay, do we have Patricia or anyone from Saskatchewan that wants to participate in the closeout? No. Okay, we got you two guys. Alan, I just talked about your grasshoppers, if you heard that, as maybe there's an RNA.
I think that was Anthony.
Well, that was Anthony? Okay.
Yes.
Do you have grasshoppers?
We do.
Okay.
We have it all.
Anyway, what do you got on your napkin for today? Here, you can have the mic.
I have a mess. No. No, actually, great day, great canola week once again. And I think one thing that I picked up here today was somebody had mentioned GM canola wouldn't have happened if we hadn't have had private-public partnerships. And that, to me, was probably the last big transformative move in canola production. So as this was what I scratched down earlier, right? So I was looking at it and going, okay, how do we engage maybe the public to work with potentially, as I listen to the startup people here at the end, how do you work with them? But maybe we need to form some more public-private partnerships to advance the next great move in canola. Yeah. So make it transformative, and that would be great. But the voice is getting screwed up again today. Well, every day. But anyhow, that to me is something I think from being on a canola commission, maybe we need to discuss that at board level, say, okay, this is where our research priorities have been. Do we start looking a little bit bigger and a little bit beyond what we've traditionally been doing? So that's all I've got.
Thanks, Alan. Always hard to follow up on your comments, but I think I've been coming to Canola Week for 8 or 10 years now, and I always find this last day one of the more challenging days, but also one of the most interesting days. Challenging because I don't have a science background, so a lot of the technical details are above me, but it's always interesting because you hear a lot of the new stuff, like today's the presentation from the innovators. So that's really good. But some of the items that really caught my interest was the drone work with the hyperspectral sensors. That was very interesting because, a lot of farmers are getting into using drones, and if this is another tool, and if it can detect clubroot, maybe it can be used for other diseases or other problems that we have in the field in producing canola. Presentation about verticillium stripe. It's good to hear that there's some work going on. Sounds like there's some varieties that are out there that show resistance, and we'll just have to kind of get some more information on that, and then hopefully we'll have some varieties coming out in a couple of years that have really good resistance. And finally, I guess, just this, in the innovation thing, one of the things that really caught me besides the flea beetles was plant-driven agronomy. That really blows my mind, that where the plants will tell you, I have a problem. Now, it's like going to the doctor saying, doc, my hip's hurting. What's wrong with it? Now we're going to have the plants telling us, hey, we're hungry, feed us, or we're sick, give us some medicine. And that's going to be really interesting development, I think, going forward.
Thanks to both of you guys. Charles is a director with Manitoba Canola Growers, and he's filling in for Jason, who had to leave early. So thanks again, Charles, for coming up. Thanks, Alan. I've got 2 minutes to talk, and then we're done. So I just wanted to thank everyone who came in person and for everyone online. Thanks a lot for participating and registering. I want to thank the large committee of people who put this event on. The names are up on the screen. I don't have them on my sheet, so I'm not going to read them. Also, a big shout out to Aaron and Caitlin with Spark. I came on the planning a little bit late, and so it was really nice to have Caitlin and Aaron's legacy of helping with this event for so long, so many years. So thanks to both of you back there. And of course, thank you, Sally. Sally's been chairing this for a number of years, so it was really nice to have Sally, who I could lean on for direction. Okay, what else do I have here? Oops. Let's see. This is. Thank you to the staff from TCU Place and the PR Productions. Thanks, guys, for making this all work relatively seamlessly on the AV side of things, which is really nice. Most of these presentations they were all recorded, but most of them will be put on the Canola Council YouTube site, which is just youtube.com/Canola Council. Sometime in the new year, they'll be put up if you want to watch them again. And one final reading of the sponsors. BASF, Bayer, and Corteva, they were the top sponsors. I can't remember what the categories are. And then we have AgWest Bio, and then the 3 provincial canola grower associations, so big grower support.
through Alberta Canola, Manitoba Canola Growers, and the host province organization, Sask Oilseeds. NYA Ventures, which is Alison's company, and Alison also sponsored part of this afternoon, well, all of this afternoon's program. So thank you so much, Alison. What do we got here? We got the genomes. We got Genome Canada, Genome Alberta, Genome British Columbia, Genome Prairie, and Ontario Genomics. We've got Canterra Seeds, Brett Young Seeds, and DL Seeds. Well, we also, on the grower front, we have CCGA, Canadian Canola Growers Association. We have FCC, Syngenta, Coke, Agronomic Services, Blue Sun, Innovation Saskatchewan, and MNP. Anyway, thanks to all of the sponsors. Thanks to all of you for coming. Safe travels home. Bye-bye.